All right, welcome back, fellow scientists. So we're going to dive right in. We are still describing the 3 steps of cellular respiration, right? So if you'll remember, the first step was glycolysis, and so we looked at that right here. So in glycolysis, we took sugar. Right, we started with glucose, we invested a little bit of activation energy, we used 2 full batteries, right, and then we got 4 full batteries out of that, and we got 2 full baskets, and then we made pyruvate or pyruvic acid. So here, this is the goal, right here is ATP, so that's good, that's usable, the cell is very, very happy. But these baskets, they have to go into what we're going to talk about today, the electron transport chain, to actually make ATP, because if you remember, our life equation for cellular respiration, energy is a product, and that's the goal, and that's the whole reason why our cells do this, is to get energy. So that's glycolysis, and then that pyruvate, then that fed into the Krebs cycle, right? And you'll notice we have 3 carbons, and then we have 3 CO2 that leave, right? So things are broken down. As things are broken down, other things are built up. So notice we just make one little molecule of ATP for every turn of the cycle, which, by the way, the cycle goes around twice for every glucose that goes into glycolysis because it makes 2 pyruvate. So ATP, we have one of those, right? But then we have 4 NADHs and we have 1 FADH2. So we have 5 full baskets that we need to do something with. So the electron transport chain is why the cell makes those baskets. It's what those baskets do, or it's where those baskets go. So I kind of cheated a little bit because I want this video to be shorter rather than longer. So I drew this, and these are just a series of proteins, right, in a membrane, in a phospholipid bilayer. So pause it and then recreate this in your notebook, and then unpause it, and then obviously we'll continue. So go ahead and do that. All right, so electron transport chain, this occurs in the inner membrane of the mitochondria. Inner membrane of mitochondria. So that's this thing right here, right? You guys all notice that's a membrane, phospholipid bilayer. So this is the inner membrane of the mitochondria. So that's inner membrane, IM. Right there. Okay, so here's the process. It's just a series of proteins, right? And we'll name these proteins as we go along. Not the actual names, but names that will make sense to us. So we're going to take our full basket, NADH, right? What is it full of? Right, electrons, which are basically pure energy. And NADH is going to drop off its electrons. We'll call this pump 1. So this is P1, pump 1, right? And it's going to drop off its electrons. So now our full basket becomes a Yep, that's right, empty basket, so NAD plus, and then that empty basket goes back to glycolysis and back to Krebs, and it's the circle of life. Thank you very much, Lion King. So this electron then gives pump 1 the energy to do this. It takes a hydrogen ion and it pumps it from the matrix to the inner membrane space. All right, that's key. That's gonna have big consequences. So then this electron goes into transport protein 1. Right, so this is TP1, and all it does is it's just a bridge between pump 1 and, what do you think? Yeah, you guessed it, pump 2. So pump 2 gets the electron. What do you think pump 2 does with that electron? Well, the electron energizes the pump and it pumps the hydrogen ion across, right? So then the electron goes into transport protein 2, which is just a bridge, not a big deal, right? And so then it goes to, what do you guess? Yep, that's right, pump 3. And this electron, almost all out of energy now, almost not able to reduce anything. So this is gonna energize the pump one last time, energize another pump, and it's gonna transport a hydrogen across the membrane into the inner membrane space, right? So let's go ahead and let's kind of make sure that we have a right. I know you can't do this, but I'm just doing this for the video so that you guys can see the effect. So then we're gonna have another H plus here, right? Another H plus here, another H plus here. Okay. So then this electron, right? Oxygen. What do we do with this electron? Well, oxygen, O2, comes in at the very end of the electron transport chain, right? And it combines with 4 hydrogens, just like this. So hydrogen plus oxygen, you guys guessed it. You're right, it makes water, H2O. right? And this oxygen, this is huge. We're going to talk about this in fermentation, but this oxygen accepting this electron is the whole entire reason why we need to breathe and why we die if we don't get enough oxygen. Because this oxygen, if this doesn't pick up this electron, then everything gets all messed up and our cells don't get energy, and if our cells don't get energy, then our cells die, and if our cells die, then we die, right? So this little thing right here, to pick up this little thing right here, that's the whole entire reason why we have our respiratory system and our circulatory system and hemoglobin and all that very, very complicated stuff is because of this little thing right here. All right, so this oxygen combines with these 4 hydrogens to make H2O, and actually it makes 2 H2O because notice we have 2 oxygens. So let's update our little picture. So we got rid of that hydrogen, that's now in water. We got rid of that hydrogen, we got rid of that hydrogen, and we got rid of that hydrogen, right? So now what we've done is we have a hydrogen ion gradient. We have lots of hydrogen ions up here, and we have very few hydrogen ions down here. By the way, I should probably label this diagram just so everyone is kind of orientated, right? So this is the matrix, right? And then this is the inner membrane space, IMS, inner membrane space. So what do these hydrogen ions want to do according to diffusion? Right, well, diffusion is the movement of a substance from an area of high concentration to an area of low concentration. Now help me out, can these ions cross this membrane by themselves? No, because this is hydrophobic, and these ions, they're charged, they're hydrophilic, so they can't just go across the membrane by themselves. They need help. They need to do facilitated diffusion. Remember that from chapter 7? So the protein that facilitates their movement or their diffusion is called ATP synthase. ATP synthase, right? And so these hydrogen ions, they want to flow back across the membrane into the matrix, and they have to go through ATP synthase. ATP synthase, in my opinion, is the coolest protein in the whole entire world because it acts like a turbine on a hydroelectric dam. Right, being from Washington and having lots of hydroelectric power, right? Most of you probably been in the Grand Coulee Dam and got the tour and everything like that. So you know that what happens, what a hydroelectric turbine does is it spins as water flows through and it makes electricity. That's what ATP synthase does. As the hydrogen ions flow through, the hydrogen ions are kind of like water, right? ATP synthase is like the turbine. Okay, so that's going to spin, and that's going to generate enough kinetic energy to take an ADP, obviously, plus PI, plus inorganic phosphate, and make it into ATP. And that, ladies and gentlemen, right there, that is the whole entire goal of cellular respiration, is to make ATP. Okay, so now that we've described it in pictures, now we're going to go ahead and we're going to describe it in words. Right, so let's start with black. So step number 1 is going to be right here, and I'll have to admit I kind of cheated a little bit just because I don't want this video to be like 20 minutes long. So step number 1, pump 1 takes electrons from NADH and pumps hydrogen ions from the matrix right here into the inner membrane space. All right, so that's step number 1. Step number 2, right, occurs right here. So transport protein 1, so that's step number 2. Transport protein 1 transfers the electron from pump 1 to pump 2. And it also accepts electrons from FADH2 and gives them to pump 2. So I forgot that. It's a good thing I made this in advance. So let me go ahead and let me draw that in. Line color. Nope, there we are. We want that. So another thing I forgot to draw this. So we have our other full basket, FADH2, right? And that doesn't go to pump 1, but it's a different basket, it's a different molecule, right? So FADH2 can only drop off its electrons at transport protein 1. So then it drops off its electrons and then it turns into an empty basket, FAD+, just like that. There, now our drawing is complete. Hopefully I didn't forget anything else. So now we have our third step. So our third step obviously involves pump number 2, right? So I tried to, even while I was typing, I tried to be really short so that you could be really short in your notes. So pump 2 does its thing. It does the same thing as pump 1. It takes a hydrogen ion and it pumps it across the membrane into the inner membrane space, right? So that's 3. And then step number 4, right here, of course, involves transport protein number 2. So transport protein number 2 does its thing, same thing. It's just a bridge from pump 2 to pump 3, right? And then we're going to have, let's do, where are we? Let's do purple. So this is step number 5 right here. So this is pump 3. So step number 5, right? So pump 3 does its thing. It takes a hydrogen ion from the matrix and it pumps it across into the inner membrane space, right? Active transport because it's requiring energy. And it also gives up its electrons to oxygen, which combines with hydrogen. So, which electrons to hydrogen and, or sorry, oxygen and hydrogen, right? Making water. That's an and sign. That's a plus sign, right? So that's number 5. And then number 6, let's go with olive green. I like that one. So number 6, ATP synthase then, is the only way that these hydrogen ions can get back across, facilitated diffusion. So number 6, they're going down their concentration gradient from an area of high concentration to an area of low concentration. Diffuses through ATP synthase, which acts like a turbine on a hydroelectric dam, and then it turns ADP into ATP. All right, so this video, you have gotten a very brief overview of the electron transport chain, which takes these baskets made by glycolysis and Krebs, and it uses these baskets to power the chain to create the gradients to make ATP, right? This is our end product right here. This is the main thing. This is the whole entire reason why we have to breathe. It's the whole entire reason why our cells are still alive and how they get energy is making ATP right here. So that's what you learned. This has been Mr. Leo. Thank you so much for watching, and I will see you on the flip side.